new chapter, Biotechnology, Principles and Processes. I hope you already heard about the term biotechnology. What is biotechnology? From the word we will get two terms. One is bio and another one is technology. So we can say that biotechnology is the application of technology in living organisms. So another one of the definition we can give related to the biotechnology. It is the technique of using live organisms or their enzymes for products and process useful to human. What is biotechnology? It is the application of technology in living organism. That may be in application of cells or maybe in parts or maybe in organism. That is biotechnology. So biotechnology is the technique of using live organisms or enzymes from the organisms to produce products and process that is useful to humans. That is called biotechnology. There are different types of biotechnologies are there. One is modern biotechnology and another one is oldest form of biotechnology. According to the European Federation of Biotechnology, that is EFB, give a one definition related to the biotechnology. It is the integration of natural science and organisms, cells, parts thereof and molecular analogues for products and services. Before I already mentioned what is biotechnology. It is the application of technology in living organism. The same things explained by European Federation of Biotechnology or EFB. There are two types of biotechnology. One is oldest form of biotechnology and another one is modern form of biotechnology. What is the oldest form of biotechnology? That type of biotechnology is mainly based on the natural capability of the microorganism. Some of the examples are preparation of wine, making curd, making bread. These all are the different type of oldest form of biotechnology. These all are microbes mediated process. Is it clear? Such a type of biotechnology is called oldest form of biotechnology. Another one is the modern biotechnology. The modern biotechnology is mainly based on the genetic engineering. It is mainly based on the genetic engineering. It includes in vitro fertilization, that is test tube baby program. Then another one is synthesis and using of gene. Then preparation of DNA vaccine, correcting a defective gene. If any gene is defective in our cell, that particular defective gene is corrected by genetic engineering process. These all are the different type of modern biotechnology that is mainly based on the genetic engineering. The first one, the micro-mediated process like making curd, bread, wine, etc. That is oldest form of the biotechnology. And another things, in vitro fertilization, synthesis and using of a gene and preparation of DNA vaccine and correcting a defective gene, mainly based on the genetic engineering. Is it clear? So next we are going to discuss about the two core techniques of modern biotechnology or principles of biotechnology. There are two core techniques of modern biotechnology. One is genetic engineering and another one is bioprocess engineering. What are they? One is genetic engineering and another one is bioprocess engineering. What do you mean by genetic engineering? Before I already mentioned, the modern biotechnology is mainly based on the genetic engineering. Even the in vitro fertilization, synthesis of gene, these all are mainly based on the genetic engineering. That genetic engineering is one of the principles of modern biotechnology. What is genetic engineering? In this technique, the genetic material, what are the genetic material? DNA and RNA. This genetic material is chemically altered and that particular chemically altered DNA and RNA is introduced into the host organisms and change the phenotype. What is phenotype? Their physical appearance. That is called genetic engineering. First of all, their genetic material is chemically altered. After that, that chemically altered DNA or RNA is introduced into the host organism. And change the phenotype of that particular host organism. That is called genetic engineering. Then another one technique, that is bioprocess engineering. That one also one of the core technique of modern biotechnology. In this technique, maintenance of sterile ambience in biochemical process for the growth of desired microbes or eukaryotic cells to produce a large quantities of biotechnological product such as antibiotics, vaccine, enzymes. Here, that particular desired microbes or eukaryotic cell is grows in which condition? Sterile ambience condition. That is called bioprocess engineering. These are the two important core techniques of modern biotechnology, or we can say that these are the two important principles of modern biotechnology. Last chapter we already discussed about the plant breeding program. So we know that the traditional hybridization program is mainly used in this method. Undesirable genes are multiplied along with the desirable genes. So the undesirable genes are very difficult to remove. This drawback has been overcome by the development of genetic engineering. Genetic engineering case, only the desirable genes are isolated or only the desirable genes are introduced into the host organism. But in the plant breeding case, along with the desirable gene, undesirable genes also multiplied. That is the reason why very difficult to remove the undesirable gene. But in the case of genetic engineering, only the desirable genes are introduced into the host organism. Is it clear? Here we are going to discuss about the genetic engineering process. It includes creation of recombinant DNA technology or recombinant DNA, then gene cloning and gene transfer. First of all, we are going to discuss about the creation of recombinant technology, or we can say that the construction of first artificial recombinant DNA. In 1972, Herbert Boyer and Stanley Cohen constructed first artificial rDNA molecule or recombinant DNA molecule. Who developed the first recombinant DNA molecule, artificial recombinant DNA molecule? In 1972, Herbert Boyer and Stanley Cohen constructed first artificial recombinant DNA molecule. How they constructed first artificial recombinant molecule or rDNA molecule? Listen carefully. For constructing rDNA molecule or recombinant DNA molecule, Stanley Cohen and Herbert Boyer selected antibiotic resistant gene, that is selected from frog. Antibiotic resistant gene. Just consider this is one of the antibiotic resistant gene. Okay, this is one of the antibiotic resistant gene. After that, they link these antibiotic resistant genes with the plasmid of Salmonella typhimurium. Plasmid of Salmonella typhimurium. We know that Salmonella typhimurium is one kind of the prokaryotic cell, or it is a bacterium. So they don't have nucleus. They have one plasmid is present. What is plasmid? Plasmids are the extra chromosomal DNA, double-stranded circular extra chromosomal DNA found in prokaryotic cell, that is called a plasmid, along with their chromosomal DNA. So first of all, they select one of the antibiotic resistant gene from frog. After that, that antibiotic resistant gene is linked with the plasmid of Salmonella typhimurium. This is Salmonella typhimurium. It's a bacterium. They link antibiotic resistant gene with the plasmid of Salmonella typhimurium. What is plasmid? It is a circular double-stranded extra chromosomal DNA found in where prokaryotic cell or bacterial cell. That is called a plasmid. So here antibiotic resistant gene is present. Here plasmid also present. Plasmid is the extra chromosomal DNA found in Salmonella typhimurium. This is another one of the gene, or we can say that this is the DNA with the desirable gene. This is the plasmid. So how can we link these two? First of all, we want to cut the plasmid. First of all, we want to cut this plasmid. How can we cut the plasmid? This plasmid we can cut with the help of restriction enzyme. What is restriction enzyme? It restricts the growth of pathogen. Actually, the restriction enzyme is isolated from the bacterium. That bacterium name is called E. coli. Restriction enzyme was discovered in 1963 from the bacterium Escherichia coli, E. coli bacteria. So, this restriction enzyme is restrict the growth of bacteria. So, this plasmid is self-replicating extra chromosomal DNA. They can replicate itself. That is self-replicating extra chromosomal DNA. So first of all, that plasmid is cut with the help of restriction enzyme. So we can call this restriction enzyme is molecular scissors or biological scissors. In biotechnology, for cutting the DNA, we are using one of the tools, that is restriction enzyme. With the help of the restriction enzymes, this particular DNA is cutting. Is it? So this is the plasmid DNA. So we are cutting that particular plasmid DNA with the help of restriction enzyme. After that, this particular antibiotic resistant gene is linked with this particular antibiotic resistant gene. This is the antibiotic resistant gene. This antibiotic resistant gene is linked with the Salmonella typhimurium bacterium, or the plasmid of Salmonella typhimurium bacterium. How can we link? For that process, we are using the molecular glue. The name is DNA ligase. What we call DNA ligase. So, with the help of the DNA ligase, this antibiotic resistant gene is linked with the plasmid of Salmonella typhimurium. So, we got one of the new kind of DNA, that is rDNA, or we can call recombinant DNA. Is it clear? How they created recombinant DNA? They linked antibiotic resistant gene with the plasmid of Salmonella typhimurium. First of all, they cut the DNA with the help of restriction enzyme. After that, that cut end of the DNA is, or that cut end of the plasmid is linked with the antibiotic resistant genes by using the enzyme DNA ligase. After that, this recombinant DNA, or we can call modified plasmid. This is modified plasmid because the reason here one of the antibiotic resistant gene is introduced or inserted. So we can call this is the modified plasmid or recombinant DNA. This recombinant DNA or modified plasmid is introduced into the host organisms. Here the host organism is E. coli. What we call? E. coli. E. coli is another one kind of the bacterium that is closely related to the Salmonella. So this rDNA is introduced into the host organism, that is E. coli. So here we are going to introduce that particular recombinant DNA. So in the case of E. coli, there is extra chromosomal DNA, plasmid is already presented here. Other than the plasmid, we are introducing which DNA? Recombinant DNA. Is it? So along with their plasmid DNA, recombinant DNA also present in where? E. coli. So the rDNA is introduced into the host organism E. coli. Why we select the E. coli? Because the E. coli is closely related to the Salmonella typhimurium bacteria. So this recombinant DNA or plasmid DNA is, or we can say that the modified plasmid DNA is introduced into the host organism E. coli. Is it clear? So this rDNA or the modified plasmid DNA is replicate using the new host DNA polymerize and make its multiple copies. So we can call this is the cloning of rDNA or antibiotic resistant gene where E. coli. This is the way Stanley Cohen and Herbert Boyer constructed first recombinant DNA molecule. So for constructing this DNA molecule, they are using many tools. First one, restriction enzyme. Restriction enzyme. What is restriction enzyme? It restricts the growth of pathogen. That is restriction enzyme. So the restriction enzyme was discovered in the year 1963 from the bacterium E. coli. E. coli, what is E. coli? Escherichia coli. From the E. coli bacteria, restriction enzyme was isolated. Is it clear? So it belongs to the class of nucleases. What is nucleases? That enzyme. The enzyme which break down nucleic acid, that is called nucleases. So this restriction enzyme is belong to the class of nucleases. Nucleases enzyme category. So what is nucleases? These enzymes can break the nucleic acid. Nucleic acid means DNA. That is called nucleases. There are two types of nucleases. One is called exonucleases and the other one is called endonucleases. So restriction enzymes restrict the growth of pathogen. It is isolated from the E. coli bacteria. It belongs to the class of nucleases. There are two types of nucleases, exonucleases and endonucleases. Nucleases means what? This enzyme can break down the nucleic acid, that is DNA or RNA. So with the help of this enzyme, we can break down the nucleic acid. There are two types of nucleases, exonucleases and endonucleases. So this restriction enzymes cut the DNA at particular site or specific site into fragments. Just imagine. This is one of the DNA strand. Here we know that many nitrogen bases are there. Guanine, adenine, adenine, thymine, thymine, cytosine. Another one, cytosine, thymine, thymine, adenine, adenine, guanine. So this is one of the DNA strand or nucleic acid. So this is the 5 prime, this is the 3 prime. Then this is one 3 prime, this is one 5 prime. Is it clear? So this nucleases enzyme can break down these DNA molecules. So nucleases means that enzyme can break down the DNA molecule or nucleic acid. That is called nucleases. In that category, which enzyme is coming? Restriction enzyme. So what is restriction enzyme? That restriction enzyme can cut the DNA at specific site into fragments. The first restriction enzyme, their name is called HIND2. What we call? HIND2. HIND2. Because of cutting the DNA, we can call the restriction enzyme another one name, that is biological scissor or molecular scissor. Every exam one question is coming. Why is it called? Why restriction enzymes are called molecular scissor? Because the DNA can cut into different fragments. That is the reason why the restriction enzyme is called a molecular scissor or biological scissors. Is it clear? I already mentioned the restriction enzyme can cut DNA at specific point or specific site into fragment. That specific site or the specific point is called a recognition site. So HIND2 is the first restriction enzyme. So this hint 2, it can cut always the DNA molecule at specific point by recognizing a sequence of 6 base pair. Example, this is one of the DNA strand. Look here. Here the base pair. Guanine, adenine, adenine, thymine, thymine, cytosine. Here also cytosine, thymine, thymine, adenine, adenine, guanine. We know that DNA is double stranded. This is 5 prime, 3 prime. This is 3 prime, 5 prime polarity. Is it clear? So this particular hint 2, that is the restriction enzyme. It can cut always the DNA molecule at particular site by recognizing the sequence of 6 base pair. That 6 base pair sequence is called a recognition site. That recognition site must be palindromic. What we call? That recognition site must be palindromic. Palindrome is a group of letters that read same forward and backward. If suppose the Malayalam, the word Malayalam, if you read forward and backward, we will get the same letters. That is palindrome. The same way here, that particular hint 2 enzyme cut the DNA at specific point. That point is called the recognition site. That site must be palindromic. Look here. Here 6 base pair. Here the 5 prime direction, guanine. Then adenine, adenine, thymine, thymine, cytosine. 3 prime direction, cytosine. Then there another one strand, 3 prime, cytosine. Then thymine, thymine, adenine, adenine, guanine. 5 prime. Which base? Nitrogen base, guanine. Here also 5 prime direction, guanine, cytosine. Here also 5 prime direction, guanine, cytosine. Which means they read the same letter. That is called palindromes in where DNA pairs. This 6 base pair we can call the recognition pair or recognition site. That particular site must be cut with the help of hint 2 or any other restriction enzymes. Is it clear? Then we already learned there are 2 types of restriction enzyme. One is restriction exonuclease enzyme, another one restriction endonuclease enzyme, because the restriction enzymes are coming under the class of nuclease enzyme. Nuclease enzymes are 2 types, exonuclease enzyme and endonuclease enzyme. So we can say that restriction enzymes are 2 types. One is restriction endonuclease enzyme, and another one is restriction exonuclease enzyme. First we are going to discuss restriction endonuclease enzyme. Just consider one of the nucleic acid. We know that this enzyme can break down the nucleic acid. Is it clear? Or they can cut DNA molecule. So this is one of the DNA molecule. In every DNA molecule, sugar molecule is present, nitrogen base is present, and a phosphate group is present. So here I am going to draw one of the DNA molecule, guanine, cytosine, adenine, thymine, then guanine, cytosine. This is the base pair. Hydrogen bond between the guanine, cytosine, adenine, thymine, hydrogen bond is present. So this is the nitrogen base pair. Is it? So this nitrogen base pair is connected with which molecule? Sugar molecule. Is it? Sugar molecule. So this structure we can call nucleoside. What we call? Nucleoside. What we call nucleoside. Nitrogen base and sugar molecule we can call nucleoside. Along with this nucleoside, one phosphate group is added, what we can call nucleotide. Nucleotide. Nucleotide means nucleic acid plus phosphate group, that is called a nucleotide. Nucleotide. Is it clear? This is the structure of DNA molecule. So this is the nitrogen pair, this is the sugar molecule, and this is the phosphate group. If the sugar molecule and nitrogen base pair is coming, that is called a nucleoside. Along with the nucleoside, when phosphate group is added, we can call the nucleotide. We know that DNA means it is the nucleotide. Is it nucleic acid? Is it clear? This restriction endonuclease enzyme make a cut at specific point within the DNA. Actually, the endonuclease enzyme can cut the phosphodiester bond. What is phosphodiester bond? Sugar phosphate bond is called a phosphodiester bond. Phosphodiester bond. Is it clear? So, this particular phosphodiester bond is cut with the help of which enzyme? Endonuclease enzyme. So, we can say that endonuclease enzyme cut within the DNA. Now, these two molecules are separated. Is it clear? Then, another one is restriction exonuclease. This restriction exonuclease enzyme remove the nucleotide from the DNA molecule. Which means this particular enzyme remove which one? This particular phosphate group from here. Which means that exonuclease remove the peripheral part of the DNA molecule. Which means they can cut at the peripheral part. Understood? So, what is endonuclease enzyme? What is exonuclease enzyme? Endonuclease enzyme cut within the DNA. Exonuclease enzyme cut at the peripheral part of the DNA. Which means peripheral part, which part? Nucleotide. They remove the nucleotide, exonuclease. Endonuclease cut the phosphodiester bond. Is it clear? This is the different type of restriction enzyme. One is endonuclease enzyme, another one is exonuclease enzyme. Restriction endonuclease enzyme and restriction exonuclease enzyme. Is it clear? Here we are going to discussing one of the restriction endonuclease enzyme, EcoR1. EcoR1. So we already explained restriction enzymes are isolated from the bacterium. So here EcoR1, that is the restriction enzyme. That enzyme is isolated from the bacterium E. coli. What is E. coli? Escherichia coli. Escherichia coli. Is it clear? So we are discussing about the naming of restriction enzyme. In every examination, one important questions coming from this part. Naming of a restriction enzyme or naming of restriction endonuclease enzyme. So I already mentioned EcoRI, that is the restriction endonuclease enzyme is isolated from the bacterium E. coli, Escherichia coli. So the first letter E representing the genus part of the bacterium. Genus part of the bacterium. What is the genus part? Escherichia. This particular restriction enzyme is isolated from the Escherichia coli. So, Escherichia is the genus part. So, the first letter is representing the genus part of the bacterium. Okay. Then, next is two parts. Second and third part is representing the species of the bacterium or species part of the bacterium. Then next letter R, that is representing the strain of bacterium. Strain of bacterium. Here the strain RY13. What is strain? It is the subvarieties of bacterium. Okay? It is the subvarieties of E. coli. That is R. R is the strain of bacterium RY13. Then 1. Roman letter 1, that is representing the first order isolation. First order isolation. Such a way that restriction enzyme is named. The restriction endonuclease enzyme cut the DNA in two ways. One is blunt end ways, another one sticky end. One is blunt end, another one sticky end. Restriction endonuclease enzyme cut the palindromic DNA. Is it? We know that restriction endonuclease enzyme cut at specific site, that is the recognition site, that is palindromic site, or we can call that is the palindromic nucleotide. So that particular restriction endonuclease enzyme cut at the center. So we will get equal length of nitrogen base pair. Such type of cutting is called a blunt end. Blunt end or blunt cut. Next one, sticky end. Sticky end means endonuclease enzymes cut the DNA a little away from the center, which means from here. If they cut from here, a hanging part will get. That is called a sticky end. Look here. This is one of the palindromic site. Guanine, adenine, adenine, thymine, thymine, cytosine, 6 base pair. So here, cytosine, thymine, thymine, adenine, adenine, guanine. Is it clear? This is one of the recognition site palindromic nucleotide sequence. Is it? So here, endonuclease enzyme cut the DNA little away from the center. This is 5' polarity, this is 3'. So automatically this part is 5', this part is 3'. So if they cut like this, what we will get? 5', 3' polarity. What we will get? G plus balance AA, T, TC. Then here 3'. What we will get? C, T, T, A. Is it clear? Balance G. Look, like this only getting. If they join these two sticky end, what we will get? Recombinant DNA. Understood? Look here, the sticky end. A core, a restriction endonuclease enzyme cut the DNA little away from the center. After that, they got the sticky end. Understood? These two sticky end is ligated with the help of DNA ligase. So restriction enzyme exonuclease and endonuclease. They remove nucleotide from the end of the DNA. Endonuclease, they cut at specific position within the DNA. They bind to specific recognition sequence of the DNA and cut the two strands at specific points. Look, vector DNA, foreign DNA. Little away from the center. Here also got sticky end. Here also got sticky end. These two ligated with the help of the ligase enzyme. So we got the recombinant DNA. So we already cut the DNA at specific points. So we will get many fragments. That fragments, everyone is not same. Is it clear? Some fragments are more size, some one is less size. Like that only. So the next step after cutting the DNA, the next step is gel electrophoresis. What we call gel electrophoresis. It is the technique for separating DNA fragments according to their size under the influence of electric field. The technique is called gel electrophoresis. With the help of the restriction endonuclease enzyme, we already got different DNA fragments. So that different sized DNA fragments is separated out from the DNA molecule according to their size under the influence of electric field. That technique is called gel electrophoresis. By using the gel electrophoresis technique, we can separate the DNA fragments according to their size under the influence of electric field. For that, we are using one of the device, gel electrophoresis device. This is the gel electrophoresis device. So, with the help of the restriction endonuclease enzyme, we cut that particular DNA fragments. After that, that particular DNA fragments are placed in where? Gel electrophoresis device. That gel electrophoresis device is connected with the electric field. For this process, we are using one of the gel that is called agarose gel. That is the polymer of seaweed. We already know that what is seaweed. It is obtained from the brown algae. That is called agarose gel. So here, the medium or the matrix is agarose gel. So we can call agarose gel electrophoresis. That is employed to check the progression of a restriction enzyme digestion. Clear? So this is one of the gel electrophoresis apparatus. Here, this particular apparatus is connected with the electric field. So we have placed that particular different fragment sized DNA molecule into the well. These are the wells. DNA is actually the negative charged particle, so it moves towards the anode. Because we are providing the electric field, is it? This process is take place by the help of electric field, under the influence of electric field. So here we are applying the electricity. This particular negatively charged DNA molecule is moved towards the anode part. All the DNA molecule is not moved towards the anode part. Which one will move? DNA molecules are separated according to their size through sieving effect of agarose gel, which is a polymer from the seaweed. Smaller sized fragment move further, which means small particles of or small fragments of DNA is moved towards the anode part, and the bigger size is undigested. The digested one is which one? The smaller one. That small one is moved towards the anode part, and the undigested one is placed in which part? Cathode part. Is it clear? Look, this first line is undigested one. Bigger sized one stay back there, and smallest one move towards the anode part. The second line to fourth line, all that one is moved towards the anode part. In this way we can separate the DNA fragment. So in this way we already separate the DNA fragments. How can we observe these DNA fragments? For visualizing these DNA fragments, we are using the stain ethidium bromide. This ethidium bromide provide a bright orange color. Directly they are not providing the bright orange color. First of all, that particular DNA molecule is applied ethidium bromide and exposed to UV radiation. First step, we already stained ethidium bromide. Which one? DNA molecule. Followed by exposure to UV radiation. Then only visualize the DNA molecule. The color become bright orange color. Look here. These are the DNA molecule. So after staining the DNA molecule with the help of the ethidium bromide, we can visualize the DNA molecule. After that, the colored part of the DNA molecule is separate and extract out from the gel piece, from the agarose gel piece. That is called elution. Then followed by the cutout gene of interest from source of DNA, and a cut vector are mixed and ligase is added. It create recombinant DNA. So after the elution we got what? That cut end of the pure DNA or the purified DNA. That purified DNA is mixed with the plasmid vector of another one of the bacterium with the help of the ligase. We got the purified DNA. This purified DNA is used to construct the recombinant DNA by joining them with the cloning vector. For joining which enzyme is helping? Ligase enzyme is helping. This is gel electrophoresis. What is gel electrophoresis? It is the techniques for separating DNA fragments according to their size under the influence of electric field. That is called gel electrophoresis. For gel electrophoresis, which medium we are using? Agarose gel. What is agarose gel? It is the polymer of seaweeds. What is seaweed? It is the brown algae. DNA fragments are negatively charged, so that particular DNA fragments are moved towards the anode. The digested one move towards the anode, and the undigested one stay back where? Cathode position. The bigger size one stay back in cathode position, and smaller one is moved towards the anode part. After that, the visualizing that particular DNA molecule, we are applying the ethidium bromide followed by exposure of UV light. So we will get orange bright colored DNA molecule. That orange bright colored DNA molecule is separate out from the gel piece. That is called elution. That is the purified DNA. That purified DNA is used to construct the recombinant DNA by joining them with a cloning vector. For joining which enzyme we are using? DNA ligase. Understood? Next we are discussing about the cloning vector.